All right, everybody knows.
Can you switch into presenter mode on Zoom? It's the slides mode.
It's it's fine here in the room to show on the big screen.
Maybe in the display settings set to mirror so that both displays are the same.
This is a PC, and I'm at a complete loss.
This looks great.
Yeah, all right,
fantastic.
We did it. Okay. Well, sorry for some logistical hurdles to overcome. Good morning, everybody. Thank you for being here at this early hour. This is the session of the interest group for physical samples and collections in the research data ecosystem. My name is Kerstin Lehnert, and I was one of the co-founders of this interest group. And online we have Jens Klump. Jens, you want to say hi?
Hi, you will see me maybe at some stage.
See you later. And we have three other co-organizers of the session sitting here in the front row, who you will see later in presentations. So I hope, nope, that one didn't work. That one doesn't work. How do I get it to move? Okay. You heard the session is being recorded. For those online, please mute yourself when you're not speaking. If you have your computer or laptop or tablet, whatever, please fill in your attendance. We really like to know who was here, and would also like you to sign up for the interest group in case you are really engaged in the physical sample and collection world. And for questions and comments, use the chat or the session document, and Jens will monitor that. Whoops, this is very hard to see. Okay, just a quick introduction to the interest group for those who, actually, a quick question. Who is in a session of this interest group for the first time? Wow, that is amazing. It's great, because that shows there is an increasing, I think, awareness of this issue of physical samples, how they are part of the chain of provenance for a lot of the data that we're dealing with. And it's also really a cross-disciplinary topic. And that has been sort of the driver for this interest group at the very beginning, that we wanted to engage and establish a dialogue across domains for those who deal with physical samples, and come up with more consistent procedures, processes, ways of documenting the samples, identifying samples, and so on. So those are all the topics that we have listed here, which over time we have been addressing. The group started in 2017, so just at the half point, P10, we're now at P20, and has been meeting really consistently across all the different plenaries that have happened since then. And it has addressed a lot of topics. It's best is to go to the webpage of this interest group to see everything that has happened. So again, here is the link to the Google Doc. For any notes, please feel free to add to the notes, and again, sign up so that you can become part of ongoing notifications and planning of future activities. In this particular session today, we want to focus more on expanding into new domains. Something I haven't said before is that really the effort came out of the earth sciences. There was a group that really thought about samples and then ran into other domains that had similar problems and said, okay, let's get this group going. And over time, it has expanded into some new domains. We have archaeology here. We've had material sciences come in, biology, definitely. But there are a number of other domains, or quite a number of other domains, that we are inviting to participate in these activities. So what we want to do today is talk a little bit about this catalog of groups and projects that deal with very different aspects of the physical samples in the digital data ecosystem, be it identifiers, be it metadata, and so on. We're really trying to establish just awareness of what's going on in the community. And we want to, at the end of, towards the end of this session, we really would like to have a discussion on what's the best way to further support interaction coordination among all these different activities that are going on, because we're meeting twice a year with this group, but that's, I think, not enough to really foster more of collaboration across these domains. We will provide an opportunity to give some updates about existing samples projects, and I'm really glad that, you know, last minute we're getting a few more in the room who will contribute to that. And then a major part of this session will be what is called a deep dive to look at biospecimens in the life sciences and how their activities could potentially be supported through a working group within RDA or as part of our interest group. So following just that overview, this is the agenda. So just the things that I already talked about, the lightning talks that we have, but I think we will have a couple more lightning talks, which is great. Okay, but with that, I just, this is an older slide that I had in a talk, and I just wanted to emphasize with this, this was at a meeting of a cultural heritage collections group. And I tried to really say, looking through the list of topics that they were addressing, well, this is all the same that we're addressing as well in our world of earth science samples. And so it is a real indication of how common the issues are. So Jens, you want to take over to do the review of the catalog?
Yes, I think, but how do we do this best? Do we, because
I
the
need to
spreadsheet,
probably
maybe stop sharing and I start sharing my screen?
What happens if I click on this? It doesn't seem to do anything. Then I will have to stop share. And okay, you would be able to take over, hopefully.
I think I can.
All right, good. Yes, it's here. It's
okay. So in over the last year and in preparation for the session, we've collected a couple of names and links and people of groups involved in physical samples and their identifiers. And some of them are in the room. All of them?
Jens, maybe you can enlarge a little bit.
I'll try that. Let me see how I do that. Oh. I think this is the really relevant part.
This is great.
So are there people from these groups in the room?
Yeah, we have.
Yes.
Three hands up.
I mean, we will have the lightning talks anyway, which will talk about some of them, like the IGSN, which is about identifying samples. Kerstin, will we hear about iSamples and sampling nature?
No, I hadn't planned on giving an update. I think with all that list, we should discuss about some sort of ongoing webinars to inform people. But no, I hadn't.
Well, I'll put the link into the chat for people using the chat.
Yeah, that would be good. I can just say iSamples is a project funded by the US National Science Foundation. It's called the Internet of Samples, and it's building cyber infrastructure, basically containerized software packages that help local sample management that communicates with a central instance for aggregating metadata and making those accessible as machine readable metadata. It has worked a lot on building a common metadata schema across domains. So if there's interest, we can learn more about that. I reported about it in previous sessions, so that's why I didn't include it.
Anybody else in the room?
Walter was. You had raised your.
I'm from Disco. Disco is a European infrastructure making.
Walter, could you speak into the microphone? Because people
yeah.
online can't hear you. That includes me.
Do we have a microphone somewhere?
I don't see it.
That's interesting. So anybody who speaks will have to come up to the computer.
Okay, here's better, right?
Yeah.
So Wouter Elling from Disco, research infrastructure in the making in Europe for specimens in museums and other facilities in Europe, and that contains basically all specimens related to biodiversity and geodiversity.
Great.
Thank you.
I could just say a quick word. ESIP, I'm not sure everybody is familiar with ESIP, the Earth Science Information Partners Federation in the US has last year or 2 years ago established a cluster on physical sample curation that is quite active. It's open to anybody to participate. It's not US focused. So we can provide this. Well, we have provided the link, so that's okay. We will hear about Astromet a little bit, and I think we're going to have something about the cultural heritage as well later, so that's great. I think that's basically it. So I think it's the Jens, go ahead in what we're going to do next with this.
Well, I think the next thing would be to hear if there's anything we've missed. I'm sure we have, because we've seen quite a bit of growth and activities in this field. So we would be really interested if anybody knows about other projects, other groups that are active in developing principles around physical samples. And even if you're not a member of that group, maybe just as a pointer for us to follow up.
Does anybody have anything to add already?
I could.
Excellent.
I could add that the Daphne for NFDI project, that is a project in the German NFDI, the National Research Data Management Initiative. And Daphne stands for Data from Photon and Neutron Experiments. We are right at the beginning to look into how we're going to deal with samples. So we will, in the next face-to-face meeting in April, we will sit together and discuss that.
Awesome.
Great.
That's the new domain. Anything else? Oh. No, Jens, you wanna? Yeah.
Maybe the Global Genome Biodiversity Network, GBN, they have samples. That's maybe some an organization to get in contact with also.
I sort of a little bit heard about the digital extended specimen. Is that something that we should be listing here? Could do that as well. Oh, okay, okay.
I don't know if you want to add our bioschemas for samples, specifically in the biosamples.
Yes, yeah.
I mean, I'm going to talk about it later.
Oh, okay.
So, but
yeah, in order to keep the list.
Yeah.
Sorry, which one was that?
Bioschemas, yes, that's Sarah here.
Oh.
Yeah.
All right. I mean, the list. Yeah, please.
Yeah.
We have more.
Yes, hello. I came here a bit by chance because our room was full. But in fact, we are interested in biological samples. And now, okay, I'm a emeritus researcher, and soon I'm going to finish my work. My name is Thompson. I'm from INSERM in France. But we are in the editorial board of what is called the Open Journal of Bioresources. And in this journal, you can submit collections of biological sources and have a description. So that will be enable you to have reference for your samples. So I can add the name of this journal. So
that would be great. Yes,
okay.
just to...
But that's only biological samples, but it's human samples, samples from animals, and also now from agriculture. So I think that would be of interest for those working with
I think it's
samples.
a great example for how to publish samples and make information accessible.
And it's really very easy to publish in this journal. You have a special scheme. You just have to answer the questions and be concise. And it's small articles of about 3 or 4 pages, but you have a reference and it's open, and that's it.
Great. Thank you.
I'll send the data to you.
Okay, excellent.
Thanks.
Don't fall over the cables. It's a little... Yes.
And we have a hand up here online from Richard Ostner, please.
And then we have another hand up here as well in the audience.
Excellent. So Richard first.
Can you hear me okay?
We can hear you, yeah.
Excellent. I'm Richard Ostler at Rothamsted Research in the UK. So another one to add to the list is the EBI biosamples, I think. I'm not involved in biosamples, but a few of my colleagues have used it for describing life science samples, particularly reference samples like genomes, that sort of thing. I'll put a link in the chat.
Great.
Yes, great. Thank you.
We have somebody coming up to the front in order to reach the microphone.
Yeah, sorry. So I just discovered this meeting, so that's why I'm a bit late and everything. We are launching a new work group called the Fairtracks work group, which is connected to the Fairtracks project, which is about biological data and some type of data which is used for genome browsers, but also used for, I don't know if I can show the website perhaps, but that name possibly. You're correct. Okay, sorry, no, I can't.
Yeah, no, this is just shared.
Sorry,
Australia.
sorry. Yeah.
We can't
cannot,
get it in.
no.
If you put it in the chat.
Yes, I'll do that.
That
Yes.
would be great.
So what we are trying to do is to collect metadata from very different sources according to very different schemes. And one of the things that then we have tried to provide some sort of common way of specifying is then samples, but we're not experts on this at all. So we would very much like to be a sort of adopter of this, or more downstream user, not providing samples, but using this for metadata.
Yeah, I like that.
Yeah.
I mean, to have these use cases where you really have an application of what we're trying to achieve. That's great. Thank you. So, Jens, you want to wrap this part up?
I think, yes, we have a lot of new leads to follow up on. This is a great expansion of the scope. This is really welcome. If you remember anything else, come across anything else, let us know by email or adding it to the notes now. Not too late, because we will be wrapping this up. The notes will stay around forever, but maybe nobody will look at them. But in the next the next few days in the follow-up, we will. But email is also great. So on the chat, there's also a pointer to the BBMRI, Eric, which I will also add to the list. Thanks, Omar.
Oops, doesn't come up. And now I'm sharing mine again.
Yeah, let me stop sharing so we can go back to the big screen.
So I am not good with PCs. Now I'm not on the right, you know where, which one it was.
Remember, yes.
Oh. Oh, okay. Oh, I thought it was still open somewhere. Okay. Now we need to move to where we were. Is it in the correct mode, Jens?
It is.
Okay. All right, so now we have a set of short lightning talks on various major systems and what's happening there. Trying to keep this short so that we have enough time later for the deep dive and the final discussion. So Jens, you go ahead with the...
Yeah, I'll then just tell you when to move the slide.
Okay.
We won't swap around. This is quicker. A very brief overview of the changes at IGSN. IGSN is the International Generic Sample Number, a persistent identifier for samples, with a system that was originally designed and conceived by Kerstin Lehnert. And then we worked on this for many years as an independent organization. But we found over the years that we were struggling to run this as a technical service for providing persistent identifiers and as a community organization. And so we ran a project called IGSN 2040 to look at how we can put this all on a sustainable footing. And one of the results from this project was the advice that we should partner with another organization with related aims. And so as an outcome of that, in October 2021, the IGSN organization entered a strategic partnership with DataCite to support the global adoption, implementation, and use of physical sample identifiers. And as an outcome of that, IGSN identifiers are now available as DataCite DOIs, and the existing handle IGSN identifiers are right now being aliased to the DOI system. So both will, the old legacy handles will continue to work, but any new identifiers will be minted as DOIs. On the right-hand side is a little cartoon showing the path of that process. We are fairly advanced now to the right-hand side, where the aliasing of the legacy identifiers is in progress, and what we will be working on is the discoverability of samples. So this means that the role of the IGSN organization is changing from operating a persistent identifier service to becoming a platform for interdisciplinary user communities to formulate standards and best practices for the identification of samples using IGSN IDs. Next slide. Oops. And so on the technical side, you can already start minting IGSN IDs through DataCite. DataCite has materials about how to do that and how to use the metadata. And the metadata is something that grew out of the IGSN DataCite Partnership Steering Group, where we developed a guideline on how to use DataCite metadata elements to describe physical samples in this initial profile form, focusing on the earth and environmental samples. And we're working with other communities, starting to work with other communities to develop similar application profiles for other disciplines. Next slide, please. So what we have seen in this work over the years, and others in the room will have made the same experience, that the need for describing samples vary across the community. There's no one size fits all. But what we have now with this transition, moving the technical services to DataCite, is that we have the DataCite catalog as a common denominator with a shared common metadata kernel. And the role of the IGSN organization will be to provide a platform for communities for developing common profiles, how to use that facility, but also how to portray samples on their own organizational landing pages or community landing pages with much richer metadata than you could fit into the DataCite catalog. And I'll stop there with the lightning talk. Thank you.
Are there any quick questions to Jens?
Just for the common community profiles, as I understand, these will not be represented in the DataCite metadata, or are they, or how you deal with these metadata extensions then with the metadata with the DataCite metadata schema?
So the metadata schema will be the common kernel, and then the extensions don't fit into that. That's the shortcoming of this kind of centralized system. But we still have the landing pages where we can portray the samples in a much richer way than we could do through the DataCite common kernel.
It really comes down to the landing pages where you kind of either comply with some community specific profiles or your own goals of documenting. That's all right. Then we move on to Sean.
Okay, 3 minutes. I'm going to see if I can. I'm going to set on a timer so that I don't go too long. So I'm here to talk about IGSNs for archaeology, and I'm going to mostly be doing that with my hat of being one of the leaders of the Tundra Regional Archaeology Project, a large-scale landscape and paleoenvironments project in Bulgaria that I've been working on since about 2007. Mostly, it gives me an opportunity to show nice pictures of Bulgaria, which is far prettier than I thought it was going to be when I started working there. So we do a lot of what we do is archaeological survey, line students up across the landscape, march them through, and count and collect artifacts. And I guess this is where maybe the least conventional or way of using IGSNs that I'm going to talk about is that I don't think that initially, many years ago, they were conceived of as being for artifacts, but we get a lot of ceramics, and that would be our main thing. We find them, during these survey activities. They're usually, kind of crappy because they've been plowed up and sitting on the surface for a while. But this is our basic data. Is this sort of are these clusters of artifacts, which then we do heat maps of them and define activity areas or sites based on what we find. So this is sort of the end of the workflow that you're seeing after we've done that analysis. We also did limited excavations or sondages. That's not us; somebody else did that. I just had to put a picture of a burial mound in there. Most of ours were settlement excavations, and for this we have a whole array of samples that we do. We get artifacts again, and I've been on other projects so focused on excavations where we got literally tons, like 23 tons of ceramics coming out of it. So this can be very large amounts of it. But then we also do bulk soil for various things, radiometric samples for radiometric. This is paleomagnetism, paleomagnetic dating sampling there, and then plant, animal, and human remains. And then, we analyze those with specialists, so they go to labs, they get analyzed. This is a stable isotopes analysis for diet. The same samples samples from the same skeletons were then also sent to Max Planck, and we're working with them on a genetics paper now. And then we also do a lot of environmental sampling, which is probably the closest to what longer term uses of IGSN had been. We do speleothems out of caves. That's a map of soil samples that we had a team of very talented undergraduates did. From the paleoecological samples, we do climate and vegetation reconstructions, and from the combination, and I wanted to finish like the fieldwork stuff on this slide, we bring together the artifacts and the paleoecological stuff and the soil samples all together in the end to do things like agricultural catchment analysis and productivity analysis. And then finally, I promise this is my last slide. With coming out of that project, we the team that I was working with from 2009 to 2011, we were quite dissatisfied with the state of field data collection software. So since then, the other thing I've been working on is developing field data software through the field acquired information management systems project, and we have a product called FieldMark that has now been used. The old version's been used at probably 45 projects across different domains, and environmental sampling is probably the single thing it is, even though we developed it for archaeology, environmental sampling is probably the single thing it's used for the most. And this is just a quick example of how we worked IGSN into a field workflow, where forms where you instantiate your own workflow and data model. It's domain general, the software is, and you can go through that, create a sample. You can either scan. We did two different workflows. One where you scanned a barcode that had an IGSN on it, and then that got fed into the record. This is all in the field offline. We also did another workflow where we printed it with a portable Bluetooth printer. But I think in the end the team liked this other approach where they pre-printed the labels and then scanned it in. So we've just rebuilt this software. So if anyone's interested, I don't want to turn this into a FAMES talk, but if anyone's interested, you can talk to me. Jens is involved. Others around here are involved with this, and we've gotten quite a bit of experience in integrating like IGSNs into a field workflow where you can assign them right from the beginning when you collect your samples. So that's me.
I have a quick question
Mhm.
about that. In that software,
Yeah.
how do you define who is registering the IGSNs? Do you have a fixed allocating agent, or can the allocating agent be customized?
So that was something that was handled sort of post field work. You'd get an export from the software, and Jens probably is more familiar with me in exactly how this worked. But we've got a variety of exports from this system. CSV, JSON, GeoJSON, shape files, whatever you need. And export was done with the relevant data, and then they were registered at the after there was probably, I'm guessing, and most of our projects use an intermediate data processing step or cleaning step as well. Like, it comes out pretty clean, but every project we've ever worked with has wanted to do a little bit of additional processing before they say submit to repositories or submit information to register IGSNs. Jens, if you have any more insight into the details of how that was done, you can jump in.
I think I'll just want to add that the strength of this particular piece of software is that it supports data capture offline. So there is no direct way usually to mint an identifier straight away.
Oh, okay.
We can prepare it. We have protocols for how to assign them, and that's how we preprint the labels. But then the actual minting has to happen once we're back to connectivity and the data are transferred.
Okay.
Yeah, and I guess I'm working now, like in my own on a paper on how to best stage data for being as fair as possible when you're working offline in places like this or rural Bulgaria or wherever. And it's probably about half the papers about metadata and a third of the papers about PIDs, and staging your PIDs this way when you're offline for minting afterwards. So if anyone's interested in that sort of thing, talk to me. I'm happy to hear what others have to say about it and incorporate that into what we're doing. Thank you.
All right. I'll take over quickly for this, which is, I think, a new discipline in this interest group, and those are samples in the planetary sciences, astronomy, samples that are being collected through space missions. These are images here, or small videos of the OSIRIS-REx mission that landed on asteroid Bennu and collected about 60 grams of sample a couple of years ago, and will hopefully land successfully in September this year and bring those samples back. So obviously these samples are among the most expensive ones to collect. The OSIRIS-REx mission is estimated at $1.6 billion for the 60 grams of sample. But there are other collections, and I work primarily with the astromaterials collections that are curated at the Johnson Space Center under NASA direction. This is a really unique resource for sciences, including all the lunar rock samples, soil samples that were collected by Apollo. There are meteorites, but also then cosmic dust that's collected in all kinds of forms, star dust. So a really exotic field to work in. All these samples are cataloged and numbered at the Johnson Space Center. Until just a few years ago, there was no data system to archive the analytical data that were generated on these materials. And this is where I come in, having set up this astromaterials data system that now curates these analytical data. Here it is, but I'm going to make it quick. So with a growing number, again, of sample return missions that NASA runs, OSIRIS-REx is one of them, but the next one coming up is Artemis, which is return to the moon. Mars is the one planned for after that. There has been attention paid to how we're gonna really archive and make this data accessible and how to involve the community. So a couple of years ago, we had a workshop in Arizona at the Lunar and Planetary Lab. And one of the recommendations really is the use of persistent identifier for the management of these samples. So we are now in the process of implementing this. It isn't clear when and how IGSNs may be implemented at the Johnson Space Center. We're using DOIs, ORCIDs, RORs now for the astromaterial data archive. So there will be, I think, a lot of interesting insights and experiences with creating a new archiving facility for these data. Yeah, I mean, we're implementing it right now because the archive for OSIRIS-REx data needs to be ready by fall this year. And it's an interesting use case because the mission has over 100 scientists all over the world working on these samples with a vast array of different methods. So we really need these PIDs for the samples to link all this data in this really distributed scientific program. So that was that. And maybe I can don't know now. I don't want to talk the whole time. We're lucky that Joan Murphy has agreed to talk a little bit about cultural heritage collections.
Yes,
as a new.
I find it very bizarre that I'm here. I bumped into Kirsten the other day. I'm working on the World Fair project, which is a project that's taking 11 case studies across various disciplines. So different classes. So we've got chemistry, nanomaterials, geochemistry, social sciences, population health, urban health, biodiversity, agricultural biodiversity, ocean sciences, disaster risk management, and cultural heritage. And what we're looking at is how we are implementing the FAIR principles in those disciplines with the plan to come up with a cross-disciplinary interoperability framework. So that's what we're working on at the moment, and cultural heritage is the part of the project that I'm working on. And the reason Kirsten and I connected was we've decided to focus on image files in cultural heritage and how they're exchanged. And we realized that there are so many things in common in terms of, you know, what is the object, what's the original, the PIDs obviously associated with it, the metadata. One of the things that we found in cultural heritage is metadata changes over time. How are we tracking those changes? How are we tracking the implementation of the care principles with regard to the samples or the artifacts that we have? So archaeology does actually touch very much on, it is obviously in cultural heritage, but I think the World Fair project seems to be bringing a lot of these things together, and this is my first introduction to this group, so I think I'm going to connect them a little bit more. I don't know if anybody has any questions for me, and I don't know if I said anything that was at all relevant to the group, but that's what I'm at. So.
No, I specifically think that this aspect of imagery and digital representations of the physical objects is a big topic. And from my perspective in geochemistry and cosmochemistry and so on, it's becoming an increasing demand to figure out how to link imagery to the data with spatially referenced data on samples, but also to the physical object. So I think there's a whole new space that we need to explore in this book. Yeah.
Yeah.
Alex, can you?
There.
Yeah. This is
Yeah, so I with these samples of physical objects and describing them, it's a thing that you link your data to, or a thing that you can link all your data to, and I think that it doesn't necessarily have to be a physical object. If we can find something that is a core or a primary object that we link all our research data to and describe that in a good way, that it becomes the one thing that we always find, and that'd be really good, I think. And that sort of object can learn a lot from what's being done in this sample, sample space or physical object space.
Yeah, and I think one of the struggles that we're finding, and we're very early stages of the project, is how do you define, what is the item that's being described, and how are you describing it, and every discipline has different requirements for how they're describing that item. And I think one of the things that the kind of hidden costs of this is the amount of effort that goes into this description, and how can we automate that, and how can we make things machine actionable when we do create these manifests for our items. That's something that we're really looking at as well.
Yeah, and the exercise that Jens showed with finding all these the core from different disciplines is an important exercise to maybe share how that was done, or what sort of I guess it has been shared how it was done, but I'm just not aware of it.
Yeah. So, thank you. Well, we should learn from what is being said now, what we're going to focus on maybe over the throughout the time between RDA plenaries. I really think, with the diversity of topics that's coming up and the need for more dialogue, we need to be a little more active in between plenaries. Yeah.
And if anybody wants to talk to me about the World Fair project, feel free. I'm around for a week.
Great, thank you. So as we're really starting to be a little late, I'm going to go through this really briefly. I wanted to give an update on this research coordination network that is funded by the National Science Foundation in the US. It is an effort to address more of the social, cultural aspects of the sample space in sort of complementary to the Internet of Samples project that focuses on the technical developments. And this is an interdisciplinary group that includes archaeology, biology, obviously the earth sciences, planetary sciences, and so on. What did I do this? So what the goal is, as I said, it's the social and cultural aspects, building a community that talks about the challenges in various domains. We want to think about incentives to bring people to share samples, share information about samples, but also think about governance of standards, of procedures, processes, and so on, and then come up with an outreach strategy to bring the issue of physical samples to the public and to decision makers and so on. Because we know, and I'm sure anybody who deals with physical objects here, it's still a big problem to actually store the samples, to have the physical infrastructure that preserves the samples for long-term reuse. So what we're doing is working on basically compiling metadata standards and contributing to what Jens had shown with these nice circles, policy recommendations, creating training materials, and then communicating these things. We're running workshops. The next one is going to be in early May in Tucson, Arizona, on samples in food and health. And as you see, energy and urbanization is another one, and biodiversity and climate are still to come in the next year. We're running working groups, and this is really a call for participation. If any of these working groups are of interest to you, going through the standards that exist and making recommendations, policies about sample access, about ethical collection and use of samples, A big focus right now is on uncurated collections, because we all know those collections that sit in the basement of your institute or in a lab and eventually maybe thrown out. That's a big concern in the community. And then these training materials. We're making all kinds of progress, but I think the important message was these workshops are coming up. We have these working groups. Please contact us to participate in these activities. We were kind of kicked behind by COVID, with obviously anything that does bring people together was delayed. So, and we're really looking for more participation in these groups. So that's all I wanted to say. I think we can go to Wouter now, and then we have to move on to your, where was the, do I need to stop the share to get to the other, oh. The,
Thanks, Kirsten, for allowing me this last minute presentation. I tried to make it quick, given the time. So I'm talking a little bit about some highlights on the development of our data infrastructure for collections in Europe. We aim to create a new object on the internet. We call it a digital specimen, and the digital specimen is kind of a container that will hold all the information that we have about a specimen. So information about the specimen itself, but also data that's derived from a specimen, like sequence data or trace information, and data sets or literature publications, etc., that's related to the specimen itself. These objects will have an identifier that we will have DOIs. And what's a bit unique on about these DOIs is that they will have a PIT record or FDO record, Fair Digital Object metadata that can be used by machines to discover whether the object is of interest for them without having to retrieve the actual data. This bit record has some metadata information. It has a description of the type of the object. So the schema for the object, the operations that are allowed to do on the object, etc. And it has a profile description which describes the bit record itself, the metadata fields that are there. And you see here an overview of some of the objects. Let's go to the next one. Okay, I was not yet ready here. You see that there are also in the record some community attributes, so specific descriptions of the object, so that the machine can determine that it's the right object. It will also contain an identifier for the physical object, which can be an IGSN or whatever ID is used locally to attach to the object, to the sample. The distinction between profile and type, as I already said, so the type defines the metadata, the data and the operations, and the profile describes the PIT record itself. Here you see an overview of the information that is contained in the PIT record. It starts with the FDO profile, which again has a handle, a PID that describes the metadata. in a machine readable way. So that's the first thing to access by machine, and then you have the rest of the information. This is an overview of the metadata that we include for the identifiers, the locally used identifiers for the specimen or the sample. Because these are not always globally unique, so sometimes just locally unique. We make in that case a combination with the host, the organization for which we use an ROR to identify them, and in some cases additional name of the identifier. This is an overview of the metadata, how we describe by discipline the different types of specimens that we have. This is currently so this is a controlled vocabulary which is currently under review, so it's open for comments until medio next month, and then we are going to make this final. So you see here all the disciplines that are in scope of DISCO. A couple of things to say here. We have also the agricultural samples, the genetic resources. We put them currently under botany and zoology for the animals, sperm samples on the zoology, and for the tissue samples on the botany. That could also be under a separate agricultural discipline, so that's under discussion. What's not here is biomedical samples, because it's not really in a scope of DISCO, could be added in a global overview. What's also not there is archaeology. Archaeology is a bit difficult thing for us because some of the objects in archaeology are in scope of DISCO, that are the natural materials or natural materials that have been used by humans. So it could be that in the future we will extend that to all archaeology objects.
Walter,
yeah.
sorry to interrupt you.
Yeah.
This is fascinating, but in the interest of time, I'm afraid we need to start moving on.
Yeah. Okay.
Sorry about that.
Yeah.
Will you be able to share the slides?
Yeah, I will be able to share that. So this is
Excellent.
an overview of the different metadata fields that we have as the core to all our objects, and we have some additional work going on for a standard for describing collections as a whole, and a minimum standard for the minimum information on specimens. And that's it.
Great.
Excellent. Thank you.
So with that, I hand over to Sarah and Rory.
This looks good.
It should be.
Okay. All right. Hi, everybody. My name is Sarah, and I'm going to do a brief introduction. So today we are going to be talking about, I'm from the SciLife Lab at the data center, and today we're going to be presenting together with Rory McNeil here from our space to show how both our organizations work together to provide an example of digitizing biosamples and biospecimen in life sciences. And I do say biospecimen because, unlike what we heard earlier today, is we about samples from archaeological sites and other types of specimen. We are
sorry to interrupt. Do you need
yeah.
to change the display settings again? Because we're not seeing the new slide online.
All right, just a second then, because oh, so there are a couple of different views. Yeah, there you go. So you can share this one. This one. Okay, Jens, is it better now?
Yes.
Awesome. Let's see if this works.
Thank you.
Okay, so as I was saying, we are talking about biospecimens specifically because we're in life sciences, and by biospecimen, I'm referring to any material that is taken from a biological entity, and it can be used for testing or for research, and that includes things like cell lines, tissues, organs, and this is as defined by the National Cancer Institute thesaurus, and basically the nature of these physical samples here, they pose some challenges. Some challenges are common as with other samples, like they can be lost and misplaced, or hidden in labs, or just unavailable in some form or the other. They can also be very rare and hard to get, and sometimes or most of the time they're destroyed in the process of investigation. So but with biospecimen, there's an added layer here of regulatory processes or regulatory issues that can be in connection with some types of samples, like genetic materials from individuals. And we've seen with the scientific collections becoming bigger and bigger with the technologies that we're getting every day. So single cell omics, for instance, have contributed to a whole lot of sample and sample data, associated data, and it becomes harder to handle. And these biospecimen, they play a very pivotal role in basic scientific research. So it's very important that we do take care of them and we do digitize them. And so this biospecimen digitization also offer promise for reusing current sample-based data, allowing us to do meta-analysis of multiple data sets, and that in itself promotes interdisciplinary research, and we can avoid things like duplicate efforts and consuming more of the sample and spending more of our time and effort and money into it. So digitization also offers that sample an extended lifespan, and and that goes beyond the institutional boundaries. So we've seen a lot of examples of collaborations in national and international levels, and that is quite common nowadays. So we need to kind of offer a way to get across those boundaries and address some common challenges here, like how to track samples when the local naming conventions cannot be really used. Every lab has their own naming conventions, and the sample can change the naming of the sample can change multiple times as they exchange hands or as they go through the analytical processes. But also it's important to realize that digitization offers a solution to these common issues, that to allow verification of the samples, that to which inherently supports open science and open science practices. And that's the culture that challenges the current one that we have, where a sense of ownership over information and data and physical sample is quite strong, where we see many examples of samples rendered absolutely worthless because you lose the data as the people leave. So we don't want to have this, and we want to change this. And the way to do this is to, in our opinion, amongst other things, is to have persistent identifiers and metadata. And metadata requires also infrastructures that needs to be improved, or it needs to be created in some instances, but it also needs to be user-friendly and integrated in the way that the researchers currently work. If we don't make it easy enough, they're not going to go out of their way to use it. So, but we also need community for metadata, and community plays a really important role upstream and downstream. They play a role in creating the metadata and defining what attributes and what is important to have for this specific community, and they're also important downstream in terms of adoption. So with that, we're gonna have illustration of two use cases. Rory is gonna show you from an e-infrastructure point of view. I'm gonna show you a bit more on how we gather the community and work on this. So to shape the frameworks for machine actionability. So with that, I'll hand over to Rory.
Thanks, Sarah. Yeah. How do you move the slide down?
So I think I just use
this one. Okay.
This, yeah, that's just the arrow.
Okay, thanks. Hi everyone. Yeah, so just to kind of ground what I'm going to say, hopefully to make sense. So I'm coming at this from an e-infrastructure point of view. So there's a metadata point of view. There's all different points of view, but at the end of the day, metadata, PIDs, other resources like that are going to be used in the context probably of some kind of e-infrastructure or tool. So that's if we can get our minds around that. I'm going to talk specifically about RSpace, which is an integrated electronic lab notebook and sample management system. Point one. Point two, that's designed to integrate with and be integratable with other tools and resources. So this is kind of a lot of stuff going on here, but you can see at the heart, RSpace is an ELN for experimental data, a fully fledged sample management system that's integrated with the ELN. It has a set of APIs which allow extensibility, and then it connects with all sorts of other data sources that are likely to be used, including storage facilities, including specialist capabilities, other tools like PIRAT, which is a colony management system. Cluster Market is an equipment management system. I'm going to talk about a bit about the integration we're doing with FAMES, which Sean described earlier. Also, we integrate with iRods and Omira, which are commonly used open source resources with things like Protocols.io, and then also with repositories so that you can deposit information at the end of the research cycle, as well as with things like data management plans. So that's a quick overview of RSpace itself. And we have two projects going on right now, which are specifically related to metadata and PIDs. One is Xiaoli Chen from DataSite and I have a new interest group, which had a very good meeting yesterday, and some of you were there, and apologies for any repetition, which is going to go on today. It's called Working with PIDs and Tools, which again is this theme of how do you work with PIDs in an e-infrastructure. The second thing we have is an EOS Future RDA grant, which again working with DataSite, which is around a similar theme, which is enhancing interoperability through use of PIDs and research platforms. So we've got two use cases here, and again, apologies to Sarah, because RSpace is not just confined to biosamples. So we are working on biosamples, but it's also much broader than that. So it's quite good for this project. We kind of just by good fortune have two projects. One is with a geosciences group at UIT, the University of the Arctic in Norway, where in that case they're working with rocks. What's also nice about that is that it's an individual research group. So we're looking at the researcher requirements for IGSNs. The second one, and I'm about to invite a Zoom bomber, but it's an invited Zoom bomber, so I hope it's okay, Kirsten. The other one is different. Rothamsted Research, and Richard Osler is actually on the call, and it's a pity he wasn't able to come because of last minute health issues, but he was meant to be here. But we're working with Rothamsted Research at incorporating IGSNs across an entire institution, which is another perspective and very exciting. And Kirsten, if you don't mind, I've just asked Richard if he would, since he is here online, if he would just maybe take a minute and talk very briefly about your exciting vision for how IGSNs could be deployed at Rothamsted. Richard, the floor is yours if you're able to take it.
Thank you, Rory. Would it be possible to share my screen quickly? I can, yeah, I've just got a couple of slides which I hopefully will give you some context. So for those who don't know, Rothamsted Research is the oldest or longest running agricultural research institute in the world. We've been collecting physical samples, conducting experiments for 180 years this year. So we have a lot of data and a lot of physical samples. And we have three, I think here for us, really interesting use cases, which all are broadly similar. So the first, they're all research infrastructures which are collecting samples, and crucially for us, the samples from these infrastructures are both archived, but they're also accessible to the wider research community as a resource. So the three platforms we have are something called the Northwick Farm platform, which is a
we're trying to.
That's fine. I'll
get to the sharing part.
I'll talk through it, but I can post the slides. Yeah, so the Northwick Farm platform is a dairy, cattle and sheep farm, and it's a series of hydrologically isolated catchments, which are designed as a long-running research infrastructure. It's been going since 2010, and we collect a lot of routine and ad hoc samples from this. So
excuse me, Richard.
Yeah.
Yeah, I think you should be able to share your presentation now, your screen.
You should be a co-host now, then, right?
Perfect. Can you?
Not yet.
Not yet.
It says I'm sharing.
It's sharing in Zoom.
Oh.
Not here. Okay.
Because you're viewing a different screen.
Yeah.
So it's asking me to sign in to Zoom. So I'll just try doing that.
There's still a lot to manage on this.
Oh, it's the screen thing. That was...
Oh.
Sorry, it's with the technician.
Yeah, I'll carry on talking.
Yeah, I think we're otherwise losing too much time.
So the Northwick Farm platform is our first example. So the people on Zoom will be able to see the slides, I think, still. So the second example is our insect survey. So it's quite different. It's been running for 60 years. It's a network of light traps and suction traps across the UK. So a suction trap is really just like a hoover that sticks up in the air and sucks insects in as they fly over. We have about 300 locations, and samples are collected as a kind of an insect mush every few days during the season. So we've got several 100,000 insect samples going back 60 years. These are routinely analysed to see what the contents are, but we only are interested in a few species there. So there's still a lot more information that we can extract from these samples. And then the third one, which is our crown jewels really, is the Rothamsted long-term experiment. So we have 179 years soil, grain, herbage, and fertilizer samples. And so the long-term experiments are a series of fixed plots, or permanent plots, which receive the same treatments over time. And this is quite important because it gives rise to quite different soil environments within the same geographic area. And again, we collect soil, grain, herbage, and fertilizer samples for these. And as with the farm platform, the samples and the plots are accessible to researchers as a research infrastructure. So researchers can come in, they can do direct sampling on the plots. They can request samples from the archive to do new analysis on. They can deploy sensors on the plots. And they can generate new data from these plots.
Richard, thanks. I think we only have 10 minutes left, so if you don't mind, can we go back to the presentation? That was
that's fine.
Really helpful. Yeah.
Stop sharing.
Thank you very much. Yep. Oh, there's Richard. Okay.
Right. That's quite intimidating.
Is it this one?
No, but that's one of the ones anyway. That's good. I mean, that's promising. It's not Richard anymore.
Yeah. But can you online see any presentation?
Not yet.
Not yet.
There we go. Perfect. Can you see now? No? We can see the right presentation in the room, but I'm not sure about the online.
Not online.
Yeah. Something is wrong with the...
I think we don't have. Yeah, should we just?
Yeah.
We just go ahead with it.
Was it this one?
Yeah.
Okay. Should be working.
Is that working now? Can you see now?
You're sharing a screen, yes.
Okay, great. All right. Well, I apologize for everyone for that interruption, and so I'm not going to speak for very long. But I think that was actually really useful, even in the very brief overview that Richard gave. You get a sense of the actual complexity of the actual situation on the ground. So I think that's a useful kind of reminder to all of us. So this is just a screen of how we're working to incorporate IGSNs. So the vision what we're trying to do at our space is to work with institutions like UIT and Rothamsted and say, okay, fine, how can we enable our space as a sample and experimental e-infrastructure to facilitate the kind of data collection and management that you need to do, and then also on top of that to use IGSNs and other PIDs to facilitate all the kinds of things that have been discussed today. And this is just an example of a mock-up of the ability to incorporate an IGSN, in this case an IGSN, but also any other identifier with a sample, a physical sample record in our space. And I was going to talk about the integration we were doing with FAMES, which will allow templates from FAMES with data collected in the field to be passed to our space and then back to FAMES, but I'm not going to talk about that because there's no time, so I'm not going to play the video. So the vision for our space is that our space becomes, in addition to becomes a conduit for adding value to data as data passes through the research lifecycle, to also do something similar with metadata and PIDs. So thank you, and apologies to Sarah as well.
Technical difficulties happen to the best of us. All right, so I'm gonna go through this very, very quickly. Just to mention who we are at SciLife Lab. We are a Science for Life Laboratory, an institution in Sweden for the advancement of biosciences. We started with the 4 major universities funding this, and now it's all major universities and the government. And our research community we cater to is we have a research environment with over 190 research groups. We also have infrastructures with about 1,400 unique users in nationally and internationally. And most recently also we got a lot of funding for data-driven life science with a lot of new groups joining us. And I'm at the data center where we provide infrastructures for the researchers, for the research environment, for the infrastructures and the data-driven life DDLS program to support data management and IT, fair and open science. As I mentioned, the infrastructures here, they do multiple different services. They cover all the major omics you can think of, and with about 40 different units. So one of the main challenges that we learned about here is that the most frequent scenario they face is that they get a sample from the researcher, and the researcher requests like a number of different omics to be done, so imaging, proteomics, genomics, whatever, and the sample gets shuffled between the different labs, the different units, and the data sets are lost, and we don't know who's doing what and where and all of this. I'm sure you know this story already, so I'm not going to rehash it to you. Don't want to traumatize you any further, but so the question here we wanted to know is what is the sample, where are the samples or the related outputs, and what are the sample information? Because what happens as well is that the infrastructures have to go back and forth all the time, asking for more information in order to be able to process that sample. And
sorry to interrupt. We're running out of time.
Yes, I'm getting there.
Okay.
I know.
Just one more minute, because then we need to.
Jens, you're killing me. Okay.
Yes, sorry about that. Killing me.
Oh, God. Okay, fine. So anyway, we decided to look at the sample metadata. Very briefly, core descriptors. How is the life science community describing biospecimen in that field? We looked at what is provided by the different groups and what is requested by, or what is needed by the repositories at the end point. And we came to, instead of defining a new standard, we went out and we found, okay, we also chose an e-infrastructure that suits our needs, which has its own ISATAB data model. And we also looked at other standards, and we did a crossover here, and inspired by the IGSN and the landing pages. So we wanted to create this for biosamples, basically, which ties in with what Jens described on why we are working with the data side in order to define the core kernel descriptions for biospecimen in life sciences. We settled on bioschema because, well, bioschema, okay, don't have time, so have a look at this. It's, yeah, right. And then we're working on updating the sample profile and bioschemas with a bunch of community people, or have community efforts. So we have some use cases, including the biosamples from EBI, we have SciLife Lab, we have the FairdomSeek, the ISATAB folks, DataSite, and RSpace. And with that, if you want to get involved, just if you want to get involved in updating the bioschemas for samples and defining the core kernel data site metadata, then just get in touch with either myself or Nick from Bioschemas. And you know what? We don't have time for this, so you can just log on and let me know and do your thing. You know,
scan the QR code. Thank you very much. We are desperately running out of time, but there's three short questions. So I think we have a burning topic for the next plenary. That's core and community profiles on metadata and other things that we will extract from the notes. We do need a new co-chair for this group. So you can jump up right now, and we'll make this happening. Or contact any of the current chairs, but also
especially looking for early career scientists.
Yes, yes. And it's always great to have different voices in the room, to have all these different perspectives. And if you're not yet a member of this interest group, go to the RDA webpage, find the group and join the group and work with us.
I would have a quick question, because we obviously were really packed full with topics in this session, and I mentioned earlier in doing something throughout the time between plenaries. Would you be interested in participate in any activities, webinars, discussions that would happen between plenaries? Can you just raise your hand if you would? I think it's worth doing it. Thank you. That Jens, you didn't see it probably, but that was a majority of hands up. So,
that's great, excellent. I'll hand it to Kerstin now. From my side, thank you very much for participating in this session. It was really exciting. And last word to Kerstin.
Oh, thank you. I can send you off into the break now, but wanted to thank you all for attending. I think we've shown how diverse and common the theme of physical samples is, and that there is a lot of interesting work that we can do to help many different domains properly document and identify the samples, link them to data and so on. And, please send any ideas that you have, any comments, for, ideas for these activities in between the plenaries, but for the next plenary. We had in the very beginning, I remember at the plenary in Berlin, we actually ran a workshop before the plenary to just have more time for discussion. So these are all, possibilities for the future to extend the dialogue and really dive into some of the details that we need to solve. So thank you again, and I hope that you will stay with us. And thank you for dealing with so many unexpected.
Can I just ask a quick question?
Yes.
What's the best way of communicating with this group? I think I'm keen for us to be more involved, because this is clearly becoming more.
I think that would be a great idea. I think it's clear. It's just not. Have a morning or an afternoon. You could do it much better.
Send an email to me and I'll follow.
6 months will be even more.
I'll send you my email in the chat.
No, thumbs up to that.
Perfect.
Hello, I would just like to say hi.